Before I start, two sentences. Thank you for inviting me for this wonderful movement, I will say, on alternative to animal testing. And Cliff also, sir is here because some of the data we are going to show it, we took it here. And thank you, sir. So I will be talking on the three-dimensional culture system. We developed 3D cultures with 3D bioprinting. And animals are, we know, I will skip these slides. Animals are used for risk assessment, which is a necessary mandate analysis for regulatory requirements. So whenever we have chemicals, drugs or anything, this has to be tested. Any pharmaceutical or anything which goes into the environment has to be tested in animals. So this is expensive, time consuming, labor intensive, and it cannot be equated to human physiology, ethically challenging, and most of the pharmaceutical require two species analysis in two different species, because it is already known that one is not sufficient. And finally it will be a non-primate, non-human primates, and then finally after all these things 1/3 or more than that will fail in clinical trials. So there we pitch in. And there are how this evolved as an alternative testing movement. So finally, I think the group is very familiar to all these regulatory things and history, because morning itself all this topic has been introduced. In the interest of time, I will move to the next slide. Is it possible? Is it possible means there are examples before us. So one is computer models can be used as a evidence, that is a record to be used as safe to be used in humans. And second is the MATEC epidermal or epiocular system, which is commercially available. You pay, your tissues are there in the lab, so much advanced. And then now insulin is produced by recombinant methods, so that is all possible. These are all from microbes. So this is possible. Finally, ending up in replacing, reducing, or refining the animal alternatives. Now, here we work. It might be taking around some 15 years to devise or discover a drug, formulate it, and then test in animals, and then go to different levels of clinical trials, and then get it an FDA approval and put it into market. But unfortunately, after market launching, this drug getting withdrawn by FDA itself due to complications in hepatotoxicity. There are cardiotoxicity, nephrotoxicity, but hepatotoxicity is more. That means a drug which is meant as a painkiller is having toxicity in the liver. So there is why this difference? Because all the tests are done in animals, which are rodents, and human physiology is different from animal physiology. So there is a gap. And in vitro alternatives are trying to bridge this gap by putting human cells in the in vitro platform, so that you get a relevant data in vitro which is not visible in animals. So if you ask me whether it will replace the animal experiment, I don't have an answer, but it will definitely predict that if there is any complication that might arise in humans. So there we work to bridge this gap. And we have a 3D bioprinter which three-dimensionally construct liver tissues. This is the same slide. I think I can skip this, but I would like to point here out at every level of putting a drug into market, there is a scope for 3D tissues as organotypic models. So why this in vitro, so much promising in vitro alternative, why it is not so popular? Because the microfluidic system is known for like 30 years, but nobody used, no tissue culture lab used microfluidic system. It is promising because it is not affordable. So make it affordable in a way that it can be used as an alternative system is one of the challenges. So lack of appropriate in vitro model is one thing, and incomplete representation of tissue and organ. It is only cells. So how can you represent a tissue or an organ? Sometimes oversensitive. The drug which is sensitive to cells might be not toxic to animals and humans. Then it is conventionally done in 2D monolayer. So you need 3D culture for that. and lack of validation. This is very important. Validation is the main limitation, that none of these alternatives, though promising, cannot move further. So we come here as approach with the tissue engineering. There are two approaches, top down and bottom up. Top down means you have the construct and you seed cells. Bottom up means you take cells and build up your tissue. So we are here on the right side. We started this 3D bioprinting program in 2016. We choose a hepatotoxicity test platform because the division of tissue culture from coming is offering test systems to customers. We have scaffold based and scaffold free methods. Today I am going to talk about the scaffold based system. So 3D bioprinting is a technology that can be seen in three different levels: pre-printing, printing and post-printing level. In the pre-printing level, we choose what type of tissue you want to print, what type of cells you will use to print that tissue, what is the medium, what is the design of your construct and all. So those all comes under the pre-printing. And the printing process, you have the 3D bioprinter and the method, the SOPs and all those tests that you do for the 3D bioprinting. And next is the bioreactor, which is required for culturing or maturing your tissues and to get the organ functions. And then finally, in future, we expect that we can be transplantable also. So this is a book edited by Dr. P.V. Mohan. So in that, this we have projected that when a kite model is there, we can print. We printed this construct. So it can be used as a technology that can be used to construct tissues of clinically relevant sizes. Now we are talking about solid organs, soft organs, which is very complex, and we are talking about liver. So it is very complex. Liver has lobes, lobules, and then hepatocyte. So we cannot mimic something and analyze all the functions of the liver. So we take representative functions like protein synthesis, metabolism, and detoxification. So to print anything, you need ink. We use an ink which is an extracellular matrix for the cells or the tissue. So we call it as bioink. So bioink is nothing but a mixture of extracellular matrix, the cells and its growth factor, which is very thick, viscous, and we can put it load it into a printer, and the printer will print according to a computer design. And we have to use all the evaluation processes since it is an extracellular matrix meant to be used inside human or used along with the cells. We have to go for all the analysis that one biomaterial will pass through. So we have done all the analysis of which is listed here: biocompatibility, mechanical property, printability, shape and structure, which is called shape fidelity, and clinically proven data. And the number of cells which is required is not normal to normal culture, 2D culture. We are going to a 3-dimension, so the dimension to the number is changing from 10 raised to 9 to 10 raised to 10. We can use any type of cell to culture this. There are various printing techniques. I am concentrating on the extrusion bioprinting, which is meant for high viscous bioinks to print. There are various printers available in the market. We have Region U. Okay, I will start from here. The bioink has to be stabilized after 3D bioprinting. So it is in a pre-ink format. We use gelatin as a base molecule. We modify gelatin because it is a denatured form of collagen. Mix it with cells, and then when you shine UV to that, the gelatin cross-links. We call the ink as gelma, gelatin methacrylamide. And then once it is stabilized, it is not soluble in water. So till then it is in a solution form. So next. So in the presence of UV, it stabilizes, but the problem is, once we have a large size construct, then the UV time increases. So when the UV time increases, there is always a concern that your cell is receiving toxic, UV is toxic. Right? So how can we use a already toxic and applied test system for hepatotoxicity? So we have one invention here, we have a patent on this, that our bioink is UV safe. That means we have put certain free radical scavenging system which will not affect the cross-linking process, but it will quench all the excess free radicals, so that the cells are safe. This is porous, so that it will help in and the fluid transport and the nutrient transport. We have to print and see that all the printing parameters are same. It is, when you put a filament there, up to 5 mm, the bioink is stable as a stable filament. All the parameters were optimized, and then this is the flow. You have a computer design. The feed in the bioink in the bioprinter, print it, UV crosslink, and the construct is like that. And then over the days, it will not change its shape. It should not swell. It should not shrink. And then there should not be any weight loss. So the video. Yeah. So once the construct is printed, we will look for the proliferation, the liver representative functions like albumin and urea. So that these are representative functions. Any liver function test that we will check in our blood has to be if we are able to analyze in our culture dish. So then only we can say that it is liver representing liver. This is the process how the 3D bioprinting works. The printer prints in a multiple plate, and it is a high throughput system. Now the PhD students are trained in such a way that they put a print, they give a print command, they will print 90, 100 construct in a day, because they need a lot of replicates. Why we should go for 3D bioprinting? This graph shows that when you have Hep-G2 monolayer, cytochrome P450 is not that much expressed. But when you have a 3D printed construct, the cytochrome P450 activity increased. And cytochrome P450 is the enzyme which will biotransform your drug. So for drug toxicity, this is very essential. So this is the interspecies variation. We have printed construct with human cells as well as rat cells. We can have a dose-dependent difference between the urea and albumin synthesis. I am not going into the details, but it represents that there is difference in interspecies variation. So this is the flow diagram. This is a HND slide which shows that the cells are distributed in this is albumin positive, and then finally we are intending to do an in vitro in vivo correlation. So two products are there from this. One is the bioink itself. We have invited expression of interest from the companies, and the second is the test system, which is now open to all customers, which is who would like to do a 3-dimensional construct as their test system for their drugs and molecules. So OECD also has come up with certain modification in their guidelines which accommodates in vitro system, so you don't need to do animal testing on these. So there is always been possibilities and promising things, and when we have a system, the next step will be validation. And validation, we all know, non-validated test methods are not acceptable for the regulatory agency, so we have to do that. But before we go into validation, we started a pre-validation system, because validation is a very expensive process, very expensive. So we have to first qualify that the test system is requiring validation in future. So we started pre-validation test system. We have a plan how to get it done. So we have like 14 days program with comparing with the rat data. So we have to generate so many documents as per OECD. This is as per OECD. So supposingly we have to do all these documentations. I thank the previous director, Dr. Asha Kishore, Dr. Anthony Atala, who accepted me in his laboratory for training in 3D bioprinting, and our group working specifically in 3D bioprinting. Most of these slides, bioink was done by Shiny, and these students trained and bring up data on liver toxicity. Thank you. I think I'm on time.
Yes.
Time for, yeah. So when you're making a complex tissue like PX, is it important to have multiple cell types there?
Definitely yes.
And how do you guide those cells to contain vegetarian cells to the migration behind?
Currently, without polarization of hepatocyte, it cannot perform. So hepatocyte needs to be polarized. So what we do is, our strategy is, we separate parenchymal and non-parenchymal as the cell suspension comes, and allow the hepatocyte to form spheroids or micro tissues. We have other methods to do that. I have not shown here. And then once it is polarized, we mix it with non-parenchymal. Then the cell organizes itself.
Just to add to this, during development there are some morphogenic readings. They guide the Q of these different axes. Can you add those Q or signals like molecular signals in which cell can migrate with themselves and position themselves in a particular.
Yeah. Sure. Yes, madam.
Thank you so much for such an interesting talk. What type of standardization parameters do you include for your bio?
There are many. One is, there are many parameters. Are you referring to printer parameters?
No, the bio-ink parameters.
Bio-ink parameters. So first would be cross-linking. Then rheology. We should get a shear thinning process, property established. Like if you have a cell suspension in a syringe and we load it into the bioprinter and our printing takes more than half an hour, then the cell is going to settle and few of the layers will get cells and top layers will not have any cells. So our bio-ink is fine-tuned and designed in such a way that it becomes gel at room temperature. So when you have a suspension, it becomes gel. So all the cells are locked. And once the pressure is applied, at the nozzle tip where the maximum pressure is there, it liquefies. And once the liquid flows out, there is no pressure, it sets back. This property is called shear thinning. So we have to fine-tune that in such a way that our ink is stable at room temperature and otherwise, once you print, it will flows out. And then rheology, then NMR, FTIR, all those things. That
related to that, the chemical nature of the matrix is critical.
Very critical.
So how do you have used the heterologous matrix or autologous matrix?
We have two programs. One is gelatin alone. So gelatin is soluble in water. So ideally we have to use collagen. Collagen is not soluble in water, so we took gelatin, a representative of collagen. But gelatin disadvantage is it is soluble in water. Once you print, when you add medium, it dissolves. So we have to make the gelatin insoluble. So we modify the gelatin by methacrylation. So once you print, it will be like a construct, and then we read it with UV. UV is like visible range, near to visible range. So it stabilizes. Then we can add culture medium. It will be stable for many days, even 21 days. So it is homogeneous.
Okay. Hep-G2 cells, there are about 100 clones all around the world today. If you go and take one lab's Hep-G2 cells, the cytochrome P40 profile is different from another Hep-G2 profile in another lab that you have to take. So 100 different clones are going. How do you ensure that you get consistent data all over the world? People are using different clones of these Hep-G2 cells, saying that your Hep-G2 cells represent the human hepatocytes, but they're not hepatocytes.
No.
So hepatocytes don't grow in culture.
Yes.
They are terminally differentiated cells. Your Hep-G2, you are growing in the cell. That itself is a different from it. So how do you use that for a hepatotoxicity assay? Something is already a transformed cell line.
Yeah, we bifurcate here on the cell aspect. We use primary cells as well as cell lines because when we approach companies, they said Hep-G2 is enough for their evaluation. So we chose Hep-G2 for the initial validation part. But original validation, in the real validation, this is pre-validation step. In the original validation, primary cells will be the benchmark.
I don't know which companies you are talking to, but you are completely, they are primary hepatocytes. Don't use Hep-G2 for hepatotoxicity assessments in vitro.
So we have very excellent hepatocyte isolation setup in our lab.
Human?
No.
Yeah, we want human because rat and human are not the same.
I'm on it because we have a project now. We are